Dear friends, to continue our interaction, we are going to simplify one more prime and important topic from the genetics, namely multiple alleles. Now, to understand the topic beautifully, let me divide the whole story into three parts: definition, then some characters of multiple alleles, and some good examples. Now, let's proceed. First of all, let me recapitulate here some good statements from genetics. Point number one. According to Mendelian concept, Mendel explained in postulate of pure factor that one gene has two factors, or we can say one gene has two alternative alleles. Alleles indicate forms of single gene. Now in the second statement, or in post-Mendelian concept, it has been stated that some genes have more than two alleles. With the help of these two statements, we can write here a beautiful conclusion that the presence of more than two alternative forms in a single gene are known as multiple alleles. As the word indicate, one gene having many alleles. Now, how these alleles develop? Population have most common type of wild type gene which gets mutate or modify in different directions by different means to form mutant alleles. Mutant alleles are basically multiple alleles. Here, let me quote you a good example. We have gene I in our body having three important alleles: IA, IB, and IO. These alleles control specific type of blood group in our body. IA control blood group A, IB accordingly B, or we can say IO control O blood group in our body. So one gene having three alleles. Now this is good example of multiple allelism. Now proceeding with very important part of story. Here we are going to explain some good characters of multiple alleles. Very important and significant character. Number one, you must know it that multiple alleles have same corresponding loci. Now what does it mean? Suppose we consider chromosome number 9 with proper loci at Q chromatid. For example, this is chromosome number 9 with Q chromatid of three different individuals as shown in the diagrammatic representation. Now this is the proper loci. The loci has different arrangement of alleles in different individuals. Now here it has only one gene, three alleles which control one character, that is blood group. Hope you got the concept. Now coming to the second very important character, that according to the diagrammatic representation, gamete have one, individual two, and population contain more than two multiple alleles. Now third point, multiple alleles show complete, incomplete, and codominance effect. We can explain these points in examples in a detailed manner. Now last very important character, multiple alleles violate law of pure factor, not in individual but in population level. Individual has two alleles as stated by law of pure factor. Let me show you here a diagram. This is individual, it has two alleles. Here both multiple allele and law of pure factor is valid, but in population multiple allelism completely violates the law. Now we have different examples to explain the multiple allelism concept very clearly. First and prominent example is ABO type of blood group system in human beings. Dear friends, the whole blood group system ABO is controlled by two important genes. These genes are present in two different type of chromosomes. Let us start. Number one gene involved is known as FUT1 gene. The gene is present on chromosome number 19, especially at Q chromatid. It stands for fucosyl transferase 1 gene. Suppose this is erythroblastic precursor cell. It has oligosaccharide chain present in the cell membrane. Now here, when the gene FUT express, it forms enzyme. The enzyme adds fucose sugar to the oligosaccharide chain. Now the structure formed here is known as H antigen. Now this is the primary function of FUT1 gene to form H antigen, basic structure in all types of ABO blood group systems. Now talking to the second gene called gene I. The gene I is very important gene present on chromosome number 9 at Q13.3 chromatid. It has three alleles IA, IB, IO. So this is example of multiple allelism. Now how these all alleles express in different individuals? Let me show you here diagrammatic representation. Suppose this is erythroblastic precursor cell membrane. It has H antigen. Now allele A expressed to form acyl galactosamine transferase enzyme which adds galactosamine to the H antigen and form A antigen which indicate blood group A. Now here the case B. Here the H antigen is present. B gene gets expressed to form B transferase enzyme or alpha galactose transferase which adds alpha galactose from the cytoplasm to the H antigen and forms what we call it as B antigen. Now talking to very important allele I0. Here the allele is defective. It do not forms any enzyme, so it cannot add any type of sugar to the H antigen. So we can say no enzyme formation takes place in O blood group type. It has only one antigen, that is H antigen. I hope you got briefly concept of ABO blood group. The whole ABO blood group system is controlled by one gene and it has multiple alleles. The combination of alleles in different individuals, let me show you here. Suppose we have blood group A, blood group B, blood group O, and very important blood group AB. Now possibility of allele present in different blood groups is A has homo or heterozygous condition, IAIO. It shows complete dominance. Accordingly in B and O, allele combination is like this. Now in AB, it has alleles like IAIB. How? Because it shows codominance effect. Now we can add antibodies. A has B antibody, B has A, O has both, and AB, it lacks antibody. We can say both antibodies are absent. Now here we can conclude and add very important points to the topic that in ABO blood group system, total phenotypes present are only, we can say, 4. Total alleles present are 3. Total antigens are 2. We can write here A and B. Total genotypes are, you can count 6. From blood group A, it has 2, accordingly up to blood group AB. Now here we can add total antibodies present are 2. It was brief explanation about ABO blood group system. Now jumping to the next very important and prominent, very rare example of multiple allelism. This is coat color in rabbit. Now this character is controlled by one gene having 4 alleles. So this is also good example of multiple allelism. Let me show you allele combination. If we have C positive dominant allele in population, it will form agouti or dark gray color like this. If we have one more possibility, one more allele in population CCH, it will form chinchilla, silver gray type of rabbit. Now we have one more case. Suppose CH, it forms Himalayan type of breed having white background and black extremites. Now last one is CA, it is a recessive allele. It forms pure or albino type of rabbit. It expresses in homozygous condition only. Now let us evaluate possibility of allele combination in coat color of rabbit. Suppose in case number 1, if the dominant allele is present, the rest of alleles will get suppressed. And we have combination of alleles like this. It will form what we called agouti color because presence of dominant allele. Now case number 2, if we have chinchilla CCH allele, it will form, already we know it, chinchilla color. Now very important case, chinchilla, it shows incomplete dominance with Himalayan and albino type. It results to form new phenotype called grey color. As you know that here the grey color is formed by incomplete dominance due to the interaction between two different genes. So this is detective example in multiple allelism to show incomplete dominance. Now we have Himalayan color and recessive color in the population also. It was all about coat color of rabbit. Now it has total phenotype is 4, total genotype is 10. Now coming to the last part of story, last example, eye color in Drosophila. The character of eye color is controlled by very important gene. It has 14 alleles. Let me write it here. Number 1 allele used here is capital W. It is a dominant allele. This W allele gets expressed and form red eye Drosophila. Second one is small w. It is a recessive allele. It forms white eye Drosophila as shown in diagram. Now rest of alleles lie between dominant and recessive. They interact by different means to show complete, incomplete and codominance. They control different phenotypes like whitiny, ivory and horny type of Drosophila. Dear friends, it was all about multiple allelism topic. Thanks, have a nice day. Stay joyous and jubilant.